Hello everyone, welcome to Chem 76A. Today is the third session of our labs, and in today's session we are going to learn about how some molecules interact with the light. Those are special kind of molecules, and we will learn in theory that these molecules are such that they have a chiral center. So chiral center is a carbon atom which is connected to 4 different kind of atoms. So we will learn more about these molecules in theory, but today we are going to see in practice how these molecules interact with light. Briefly speaking, what's going to happen is that when you pass a plain polarized light, it means that it's a light in which all the waves are in the same plane. When these light waves hit these molecules, the plane of the light gets rotated. Now sometimes some of these molecules will rotate the plane of light to positive direction, right hand side, and some of them they will rotate the plane to the left hand side. The molecules which rotate the plane to the left hand side are called levorotatory, and the ones which rotate to right hand side are called dextrorotatory. So today we're gonna see, we will look at positive rotating and negative rotating of isomers of same molecule. These are called carvones. These molecules are present in spearmint oil and peppermint oil. So they physically they look about the same. So I have 2 samples. So this one is a negative carvone and this one is a positive carvone. I'm not gonna open the bottles because they look about the same, and if I mix them I wouldn't know. Well, I can figure it out experimentally, but I'll show you in a while. So this one is a positive carvone, which means that it rotates the light to positive direction, and this one is negative carvone. And the technique by which we're gonna analyze these molecules, that technique is called polarization, and the instrument is known as polarimeter. Let me show you the ancient kind of polarimeter. Okay. So this is a simple old looking polarimeter. It is actually pretty old. I think about 100 years old. Not this particular one, but the technique. So what we have is we have a hollow tube here. Let me adjust. Okay. So there's a hollow tube, and inside the tube there's a glass tube. Okay, let me take that tube out. So in the glass tube you can fill your molecule. If your molecule is liquid, you can, such as any oil, so you can fill it up. If it's a solid such as sugar or glucose, you can make aqueous solution and then fill this tube up. And then you put this tube in here, and on this side, on this end, it's hollow, and there's a polarizer on this side, and that's a light source. This one is a bulb, so this side gets connected here, and the bulb shines a light from this side. The polarizer on this end cuts down all the light in different planes, and only one plane polarized light passes through it. And then there's another polarizer here, and if the molecule rotates a light to any direction, so we have to match this rotation with this rotating device. And once the plane is matched, then we see the full intensity here, and we will note down from this scale how many degrees it rotated. So this was the old way of measuring the polarization.
Hi, I'm Tom Loschiavo, the Chemistry Education Manager at Pasco Scientific, and I'm here today to talk about the Pasco Polarimeter. So the polarimeter is a device used to measure the rotation of plane polarized light by optically active compounds. So before I get into the polarimeter, I just want to tell you a little bit about what that sentence means. So assume this is a compound with a central atom and 4 different atoms attached to it. This is the same central atom with the same 4 different atoms attached to it, but these are actually different compounds. These are non-superimposable mirror images of each other. They look the same, mirror image, but I cannot superimpose the two. These are called enantiomers, and these enantiomers, these 2 enantiomers, happen to have a chiral center, and that chiral center will rotate the plane polarized light either this way or this way, depending on the compound. So what does that mean, plane polarized light? Well, I can demonstrate that over here. So I have a light source in the back. I have a polarizing film. I have a sample of water. And I have another polarizing film. And you notice when I turn the first polarizing film that the sample and the film around it get darker. Right? The water is not having any effect on the brightness of the light. But if I take that same light and I run it through a sample with a chiral center, in this case Karo syrup, then you'll notice that in this case some wavelengths of light are getting blocked while others are getting through. So now, as this is getting darker, this is getting brighter, different colors are starting to appear because of the different wavelengths of light that are being blocked or let through by that particular sample. So that's what I mean by rotation of plane polarized light. This is rotating the light, this is not, because there are chiral centers in the compound. Now what we want to do is we want to quantify that. So to quantify that, we're going to use our polarimeter. Polarimeter, simple turn on power button right here. It can be connected via Bluetooth or USB. Inside, there is a sample cell holder, and here's a sample cell. So this sample cell just has water in it, but it's a 100 millimeter sample cell with quartz ends, so it won't affect any of the light. In the polarimeter, there is a 589 nanometer monochromatic light going through a polarizing film on this end and a polarizing film on this end in front of a detector that I can rotate around just like I could rotate this around. So I have monochromatic light that's getting plane polarized, going through my sample, and hitting a detector.
Now this is a modern polarimeter, and in this device the sample goes in this chamber and the tube which holds the sample looks like this. So in this tube I have water, and please keep in mind that water is not an optically active molecule, which means that it does not have any chiral center. So this tube or water in this tube is not going to rotate the plane polarized light. And you can see here that there's a light source, and then there are polarizers here, and we can rotate just like the other instrument to match the plane polarized light. Okay. So what happens is that you fill this tube up. You can open from here, fill this tube up with your sample. If there is any bubbles, so bubbles should be in this bulge, because the light passes from here right in the middle, so in the path of the light there should not be any bubble. These Carvones molecules, they look quite similar physically. So for example, this one is negative Carvone and this one is positive Carvone, and if you smell it, they both smell minty. In fact, they are present in spearmint oil and peppermint oil. So I cannot distinguish by smell. Some people can, but to me the fragrance is exactly the same. Okay. But their optical rotation is totally different. The positive one, it's gonna rotate the light to the positive direction, or it's also called dextrorotatory. The negative Carvone, it will rotate the light to negative direction. So let's test it with the help of polarimeter. So you put your sample in here, and then you close it, and then you go to your computer and open the software and continue the experiment.